Good morning to everyone. Thank you for having me here. I'm going to quickly start off the presentation by showing you a very iconic image from the lab of Professor John van der Oost. This image was sort of the basis of how CRISPR research started here at Wageningen. What you see here are three different plates. On the top, you have a plate that's just a control, so just an empty plate for the sake of it. And then we have two plates at the bottom. On the left, we have a plate which has bacteria that have the CRISPR immune system. And on the right, we have bacteria that don't. And then we treat the bottom plates with viruses, or phages, as they're called. And you can clearly see the difference here, that the ones with the CRISPR-Cas system are more immune. You don't see these dark spots that start appearing on the plates where they don't have the CRISPR-Cas system. And these spots are appearing on the plates because the phages, or the viruses, are destroying the bacterial cells. For achieving this immunity, the cells needed a complex of multiple CRISPR proteins. And then came the breakthrough discovery in 2012 of the single protein known as Cas9, which is actually the signature protein of all genome editing technologies right now. The specialty of this protein was that it's a single protein which can be engineered to create specific and precise cuts in the genome. Just to give a perspective of how genome editing or gene editing technology is used. Just to give an analogy, so here we have a sentence, green C is native to China. And this could be, for example, the gene that we want to edit, and you see that the C doesn't really fit there. And these are basically mutations that can give rise to really deadly diseases. So with Cas9, you can you can program Cas9 to cut exactly where you need it to. It's quite precise in that sense. And then it's up to the cellular machinery. It can either put in random letters, creating mutations which inactivates the gene, or you can, or scientists or doctors can precisely provide a repair template, as we call it, which is then integrated into the gene, thereby repairing the gene. So this is why CRISPR-Cas is so popular and so exciting as a technology, because it's really easy, it's cheap, it's fast, it's robust, as well as extremely versatile. It can be programmed really easily. But of course, as with every technology, there also comes some limitations of using Cas9. So quickly through it is that the activity of Cas9 is really dependent on the cells. So in some cells it might work and in some not. You need to be sure about the target site, and they need to be available within the genome that you want to edit. Of course, how do you deliver the Cas9 proteins into the cells? And lastly, a very important one is these off-target effects, so the unintentional cuts that happen in the genome by the protein. And in our lab, we have a pipeline of how we want to sort of solve these issues, where we discover, explore, and exploit novel CRISPR-Cas variants. And I just want to give you, if you look at the big picture of CRISPR-Cas diversity in nature itself, so the most commonly used Cas9, it comes from this particular type, and we in the lab try to look at different types of CRISPR-Cas systems. I just want to give a few examples of what we are working on currently. This protein variant was in lucky coincidence, I would say, where a bunch of colleagues were looking at lactic acid producing organisms in a compost heap nearby. And then they stumbled upon this amazing organism which harbored a Cas9 in it, known as what we named as thermo-Cas9. And thermo-Cas9, as we hypothesized and experimentally validated, is extremely robust and thermostable and is active at a wide range of temperature. And most importantly, what we observed is that at a lower temperature, it is more specific. And this is really important to increase the precision and specificity of the proteins, and currently we are trying to use this in human cells. Another protein is Cas12a, and this is being already widely adopted into the genome engineering toolbox. And an important feature of this nucleus is that it's really precise and it can also be used for editing multiple genes at one moment. With that, I just want to sum up with what we are doing now in the lab. So these proteins that I mentioned, Cas9 and Cas12a, which are widely used currently for genome editing, are really big, so it makes it really difficult for delivery. And how we want to address this issue is by exploring these really small variants, which are half the size of these big proteins, and we are very hopeful that with these they will contain or have novel functionalities that we can use for easy delivery for genome editing as well as therapeutic applications. With that, I would just like to thank, of course, John and the group, as well as the funding organizers. Thank you.